What's cracking, guys? Omar Yusuf here, back with another video. In this video today, we have a very important interview all about hypertrophy, the principles of hypertrophy, as well as investigating the effects of steroids. You see, we have another interview with Alex Turner, someone who is currently getting his PhD, essentially studying myonuclei cells. You're going to understand in this interview exactly what that means. You're going to understand the principles, the mechanisms of hypertrophy, how it actually operates. And there's just a lot of misinformation out there. We had him on previously to talk about the whole natty verified movement, to talk about drug testing, drug-free athletes, how it actually works. And from that, I wanted to talk more about his actual research, what he's currently doing, because wouldn't you know it, there's not nearly as much research as people think when it comes to anabolics, when it comes to PEDs, performance enhancing drugs, what they actually do. So what he hopes to do with some of his research looking at myonuclei cells with current and previous users is quantify some of these effects and quantify when it comes to satellite cells, when it comes to potential, which would have huge implications for sport, for performance, for everyone that just cares about lifting. We talk once again about the principles of hypertrophy, so it's going to be relevant to everyone out there, but I just see honestly too much misinformation. This is not an area that I'm very well informed of myself. I see a lot of bad information, a lot of bad videos, or a lot of misinformation in the comment section. So consider this a public utility right now, an interview talking about the science essentially of steroids and the science of hypertrophy. If you like this content, make sure to like the damn video and check out in the description Alex doing his research, where if you live in Europe, you can actually help contribute to the study to further enhance our knowledge when it comes to PEDs. Anyways, without further ado, timestamps are in the description. Let's get this video started. Well, guys, we are back with another monster video. I am joined once again with my illustrious guest, Alex, who is pursuing his doctorate, essentially studying a very fascinating topic we're going to get into. Consider this a exclusive on this channel, where some of the things that we're talking about were how to squat, how to bench press, the process of muscle growth. Some of the stuff has been talked ad infinitum, but what we're about to talk about today concerning Alex's research is brand new, and there's a lot of misconceptions when it comes to steroids, the anabolic effects, studying myonuclei cells, and so I thought it'd be interesting to get Alex back on to talk about his research and how the process, first of all, how muscle growth works, how anabolic steroids work, the mice studies, human studies, and just give greater context, because once again, that whole natty verified movement, it's not an insult to it, but it raised a lot of questions, and it just showed that there was a gap of knowledge in the fitness industry. So I'm happy to have Alex back on. Alex, my man, can you just introduce yourself briefly?
Yes, I'm a second-year PhD student at the University of Brighton, which is in England, and I'm conducting a research project that is investigating the long-term effects of steroids on human muscle, and particularly interested in if muscle exhibits a memory of steroid exposure through time, and if once someone has taken anabolic steroids, you could see the lingering effects in their muscle afterwards when they cycle off, both in the short term and in the long term. So we're specifically recruiting at the moment for our research study, and we're looking for males who are aged 20 to 40 years old, who when they come to our laboratories have just finished or are on the verge of finishing an anabolic steroid cycle. They will then refrain from using anabolic steroids for roughly 20 weeks, when they will then return back for sampling, so we can investigate if their muscle on that second occasion is exhibited a memory and looks similar to the muscle on the first occasion when they just finished their anabolic steroid cycle. That's in the short term. And then we are also recruiting males who finished taking anabolic steroids 2 or more years ago, and since then have not exposed themselves to above physiological levels of androgens, so we can see if their muscle, years after the cessation of usage, still has retained a memory of exposure, or if it looks like the muscle of people who have never taken steroids before in the past. And so we're also recruiting, as a comparison group, men who don't take anabolic steroids but lift weights regularly, and also sedentary males. And as of recording this interview with Omar, we have actually sampled over 30 individuals, 6 current anabolic steroid users, 6 past, 13 guys who lift weights and don't take steroids, and also 5 sedentary males as a control group. But we are always looking for more people. We actually have a further sampling weekend on the 10th and the 11th of August 2019. We will have more sampling weekends coming up in September and October, and also into 2020. And if anyone is interested, particularly if you're a current anabolic steroid user on the verge of finishing a steroid cycle in the year 2019, or if you took steroids 2 or more years ago, then get in touch with me. We'll be able to get you involved. If you're living in Europe, we can reimburse your travel expenses, and that's something we can discuss by email if you get in touch with me. I know my email will be in the description box below, so just send me an email and I can give you some further details on the study, and then we can work out the logistics of how we can get you to get involved on one of those weekends.
No, this is fascinating because, often on this channel, Alex, we'll try and interpret research, but this is an opportunity for individuals to directly participate in information that will be applicable and useful for everyone out there, where there isn't as much information on anabolic use as people assume. And it's trying to quantify some of these things. It can enhance our level of knowledge and therefore its application in so many different ways. And so I think today, I do want to talk about what your research is on. I do want to refresh everyone, however, before we begin. And you gave a great outline for the interview today, which would be, first off, to talk about, in the first place, how does muscle grow? What is the process of muscle growth?
So obviously, when we're talking about muscle, we're talking about the muscle fibers, which are these cylinders of contractile proteins, and they slide past each other to produce movement. Now, the name of the game when we're talking about muscle growth is you want muscle protein synthesis, which means making proteins, to be greater than muscle protein breakdown. And if muscle protein synthesis outweighs breakdown for a sustained period of time, then the muscle fibers will enlarge in size. So those actual cylinders will get bigger. And that's muscle growth. That's muscle hypertrophy. Now, the key player inside the muscle fiber when we're talking about muscle growth and making proteins is the nucleus of the cell. And the nucleus of muscle fibers are called myonuclei. And muscle fibers actually by volume are the largest cells inside the human body. So unlike a liver cell, which would have one nucleus that controls that entire cell, muscle fibers actually have thousands of nuclei inside a single cellular fiber, instead of a single cellular body. And those are the nuclei responsible for making these proteins. So what you'll want to do is, by resistance training and by consuming protein on a frequent basis throughout the day, you will stimulate the nuclei that are inside those muscle fibers to make proteins. And as I said, if protein synthesis outweighs breakdown through time, muscle growth will occur. Now, there is an issue with just stimulating the nuclei that are already pre-existing in the fiber, because the proteins they make will only diffuse so far. So they will only be able to get the cylinders to a certain size. The only way the fiber will be able to get any bigger, because the diffusion distance has been maximized by that nucleus, is you have to put more nuclei into the fiber. The nuclei will have to increase in number. And the way that happens is through hard training. You will end up activating the satellite cells, which are the stem cells inside muscle fibers. So you'll activate them and they will donate a nuclei into the fiber, and then you have more myonuclei. And the more nuclei you have, the more proteins that can be made, and that fiber that was at a certain fixed size can now actually get even bigger because it has more nuclei to make more proteins. Now, the interesting thing is that, so we're looking at two things there: to increase the amount of proteins that the pre-existing nuclei are making, and also try and increase the number of nuclei in the fibers. Those are the two ways that muscle growth can occur. But the interesting thing with anabolic steroids is they actually do that just as a molecular mechanism of action. So even in the absence of any training, and we can see in studies where they give anabolic steroids to participants, the classic Basin papers and Sinha Hakim papers, which I can send to you, Oman, you can put in the description, where they give people anabolic steroids and they see, even in the absence of resistance training, that nuclei numbers increase in the fibers. So anabolic steroids increase the number of nuclei in the fibers even without resistance training. So if you're training as well, you then have two stimuli causing nuclei numbers to go up. So anabolic steroids do this because their receptor, the androgen receptor, is present inside muscle fibers. And when steroids bind to the androgen receptor, they will stimulate the pre-existing nuclei to make proteins. but they will also bind to the androgen receptor inside those stem cells, the satellite cells, and cause them to donate their nucleus into the muscle fiber. So the number of nuclei will go up and the myonuclei will go up. And the interesting thing about that is what we're coming to realize from research in mice is that once a myonuclei is incorporated into the muscle fiber, it doesn't actually disappear through time.
Yeah, and so let's pump the brakes for a second right here, because that is actually what you're attempting to study, the myonuclei, right? Can you talk about these mice studies briefly, where some people, the question has always been, right, anecdotally, okay, so someone uses anabolic steroids and they cycle off, and from anecdotal reports, people say, oh, you lose all your gains. You know, like once you go off, whatever you had, you lose it. And people have been attempting for a long time. I even saw Greg Nuckols, where the effects of anabolic steroids, he was trying to quantify in some way how much they help, right? But people have stated for a long time that essentially when you go off, you lose all or most of your gains. But you're saying that there are mice studies. Can you just break that down a little bit, how they studied it, that in fact the myonuclei cells remained, the amount that they had, or a certain portion remained after anabolic steroid use, and how, of course, this is very important when it comes to hypertrophy or just any goal that you might have in regards to training.
Exactly, that's very true, yes. So in 2013, a very important research paper was published out of Christian Gunderson's lab in the University of Oslo in Norway. The lead author on that paper was Ingrid Egner. And in that research study, what they did is they had two groups of mice. One group was given testosterone, so an anabolic steroid, and the other group was given a placebo for 2 weeks. And as expected, the muscle fibers of the mice that were exposed to the anabolic steroid increased in size. So the cylinders actually got bigger. And also the number of nuclei inside their muscle fibers went up. But what the researchers did was at that 2-week period, they removed anabolic steroids from the mice, and they were giving the mice anabolic steroids by placing a pellet into their neck that released anabolic steroids throughout the day. So at that 2-week period, they removed the anabolic steroid pellet or the placebo pellet from the neck. And they then left the mice in their cage for 3 months, which is over 10% of the mice's lifespan. And the mice did absolutely nothing. They were just there. They weren't exercising. They were just left in the cages. And then at the end of that 3-month period, the muscle fibers in the group that were previously exposed to anabolic steroids had actually shrank down to the exact same size as the control group. So they lost their gains. They lost the size of the muscle fibers they had. But the key difference that remained was the number of nuclei was still elevated by 28% in that group. And at that point, at the end of that 3-month period, when the nuclei were elevated by 28%, they exposed those mice to a resistance training stimulus. And what they actually did was a technique called synergist ablation, where they will cut one muscle off on the leg of the mice, so it's forced to walk around on another muscle. And that muscle will grow in size massively because it has to sustain the weight of the mouse so it can move around. And they exposed those mice to that stimulus for 2 weeks. And after 6 days, the mice that were previously exposed to anabolic steroids experienced a 30% increase in fiber size, whereas the mice that were previously lifelong natties only had a 6% increase in fiber size. And at the end of that 2-week period, the mice that were previously exposed to anabolic steroids, their muscle fibers were 20% larger than the mice that were previously natural. Yeah. And the conclusion of that paper was that the myonuclei that are incorporated from steroids do not decrease through time. They are retained and actually result in a high muscle building benefit to the mice in the future, many, many months after the cessation of usage, and actually after 10% of the mice's lifespan has expired. And so the conclusion is anabolic steroids enhance muscle memory, and they do this by increasing the number of nuclei. The nuclei then retained, and then therefore they result in a performance benefit. And this research group actually over the past 10 years, even prior to that study on anabolic steroids, has proven in many different instances that the myonuclei that are incorporated for muscle growth do not decrease through time. So, for example, even without anabolic steroids being involved, they have given mice a hypertrophic stimulus, so the mice have bulked up, their fibers have got bigger, but then they have then taken away the hypertrophic stimulus and they've actually tried to make the mice atrophy. So that means they've tried to make it lose muscle mass. And they've done that in multiple different ways. And the easiest way to do that is to stop the muscle contracting. And the way they managed to stop the muscle contracting was either inject it with a chemical that would block the nervous supply, so the muscle cannot contract because there's no nervous supply. They either cut the nervous supply surgically, or they do something called hind limb suspension, where they will actually just suspend the hind legs of the mouse up in the air, and the legs are not being used, so there's no muscle contraction. If there's no contraction, there's no need to make proteins inside the muscle, and muscle breakdown occurs. And in all of those instances, they have witnessed a decrease in fiber size, so the cylinders have got smaller, but the nuclei that were present prior to that have remained. And when they then put them back onto a hypertrophic stimulus, their fibers increase in size again.
Right.
And so this group is saying the molecular mechanism of muscle memory, which is just that if you acquire muscle mass prior in your life, you may lose that muscle mass, but you can actually rebuild yourself back up quicker on a second occasion than it is the first time accumulating that muscle mass, is all to do with the accumulation and subsequent retention of myonuclei, both training naturally and also if you use anabolic steroids. And it seems to be that myonuclei, once they're inside the muscle fiber, even if the muscle fiber shrinks in size and an atrophy conditioning is experienced, they're protected and they're shielded from that reduction in size. And that seems to be, based on what this group is saying, an evolutionary mechanism to try and preserve your muscle mass through time. Because let's say you could imagine a situation where food is very plentiful and you're able to eat well as a way back hunter-gatherer evolutionary times, build shelters, and be good in terms of evolutionary fitness and be able to reproduce and survive and things along those lines. But then say a famine hits and the weather turns, there's a lack of food, you might lose a lot of muscle mass in that time period if you're not eating, and you will get smaller. But then as an evolutionary mechanism, if the increase in nuclei was the thing that made you big in the past, it would make sense that you could retain that. So then when food comes around again the following season and you can get back to eating and making shelters and doing well, if you could put on that muscle mass much quicker on that second occasion, then you can get back to reproducing and being good in terms of evolution very quickly. So they're saying there's an evolutionary mechanism as to why it makes sense that myonuclei are retained inside the muscle. It makes sense because it means your fitness will increase through time. You build up your muscle at some point, something happens and you lose it, you're actually going to be able to rebuild it quicker on the second occasion than you ever did the first time round. And this makes sense anyway, as we see, because it's known quite anecdotally, if you break your leg, it's put in a cast, you're going to lose muscle mass. But then when you get back to training, you'll be able to put on that muscle and get your leg back quicker than you may have done in building muscle the very first time around.
Breaking news, Alex. Mother Nature wants you to get swole. That's the title of the video.
Yes.
Here is the question then, because you're talking about mice studies. Has there been any research done as it relates to humans? And that actually confirms anecdotally what a lot of individuals say, where they can only observe the loss of muscle when they go off, but they don't know that they still retain their myonuclei, and therefore, should they train again, or, you know, they would be able to build muscle at a much quicker pace than what they would have before. We talked about rodent studies. What are the studies? What do we know as it relates to humans specifically?
Yes. So that egg paper, that rodent study, is obviously limited because it's in mice. And some people have criticized that technique of inducing hypertrophy, synergistabulation, of cutting one muscle off to cause another muscle to hypertrophy massively because the mice has to walk around a lot, as not very applicable to humans, because that's never going to happen in the real world in a human. And obviously, when we're talking about research with anabolic steroids, ethically, it's very difficult to justify giving somebody anabolic steroids for research purposes.
Right.
So there's very limited research when it comes to giving people anabolic steroids. There's only really a handful of papers that have been published. And in some of those papers, such as I mentioned as Sinha Hakim papers, they actually do not want the guys to exercise because they just wanted to see what anabolic steroids did to muscle by itself. And there have been a sequential of papers in that regard. But all of that is in current users, and you're seeing how testosterone increases muscle in that moment. But what we're interested in now is what happens if you stop taking steroids. What happens after that time period? And there's been very, very small amount of research in that regard. There is one research. There's a few research studies that have come out of somebody's PhD thesis. Their name is Anders Ericsson. It was published in 2006 out of Umeå University. And he compared the vastus lateralis, which is the quadricep muscle, to the trapezius muscle in powerlifters who are current anabolic steroid users, past anabolic steroid users, and powerlifters who don't take steroids, and also sedentary males. And in that research, Anders Ericsson found out that the myonuclei numbers in past steroid users were still elevated. And actually, he found out in the trapezius muscle that the myonuclei numbers were higher in the group that were previously taking anabolic steroids than the current steroid users, and that was even higher than the group that were the naturals. In the leg muscle, in the vastus lateralis, it seemed that the powerlifters that were previously taking anabolic steroids had equivalent myonuclei numbers to the clean guys, and the guys that were currently taking anabolic steroids had much higher myonuclei numbers in their vastus lateralis. But this is just an indirect form of trying to investigate, do the myonuclei that accumulate from steroid usage, are they retained through time? Is it, if we look in the muscle of people who used to take anabolic steroids, can we see if they have high numbers of myonuclei? And from Anders Ericsson's thesis, it seems in the trapezius muscle, this seems to be true. That's also the piece of research Anders Ericsson was involved in that showed that in the trapezius muscle, the percentage of the nuclei inside that muscle that express the androgen receptor is actually much higher in the trapezius compared to the quadriceps. And this sort of anecdotally might be the reason why people who take anabolic steroids tend to develop very large traps, and their shoulders tend to be more 3D and well developed compared to naturals, as it seems to be that the percentage of their nuclei in their muscle that expresses that receptor for muscle growth is much higher. So that was one piece of research that seemed to indicate that previous anabolic steroids users are retaining higher levels of nuclei in their muscle. Apart from that, there's not been much else. In 2018, Mark Bell actually received a muscle biopsy from Andy Galpin's laboratory. Yep. If you go and read the research abstract, the poster that they made on that, which is available at Andy Galpin's ResearchGate, you'll notice that it's very obviously Mark Bell, even though they blocked out his eyes, and he's since on his YouTube channel spoken to Andy Galpin about the results. But actually, in Mark Bell's muscle biopsy on his leg, he was actually shown to have twice the amount of myonuclei as a healthy, normal male. And in that research paper, it said that Mark Bell had admitted to using anabolic steroids for more than 10 years. And for the past 4 years, he'd been using over 1,000 milligrams of testosterone a week. And so he could be regarded as a current anabolic steroid user. And it seems to be that his myonuclei numbers are indeed much higher. But apart from that, there's not much evidence we can go about trying to see, do past users retain their myonuclei? And there's not at the moment been a time course study where we can see the myonuclei go down in the same people through time. And that's where our research will come in.
And that's amazing because, as you said, you know, there's limited funding in so many different ways when it comes to trying to research exercise. And so people want to know answers to these questions, but there just simply hasn't been the research done yet. And that's exactly what you hope to do. What then, Alex, is the future? You're doing current research. You gave a brief overview of what you plan on doing. But what are some of the practical implications of what you hope your research will show?
So by biopsying guys just as they are on the verge of, or they've just finished an anabolic steroid cycle, and then inviting them back for sampling 20 weeks later, we'll be able to see how much muscle mass somebody loses when they go off anabolic steroids, because it's a laboratory, so that'd be very interesting. So if someone is using anabolic steroids and planning on coming off, that's just something interesting for them to know. Yep. But by conducting the muscle biopsy, we're also going to be able to see, we'd expect that their nuclei numbers would be very high at the end of a steroid cycle. Their fibers would be very big. And then the question is, when they come back 20 weeks later, how much have their fibers shrank and have their nuclei numbers gone down significantly in the short term, in that 5-month window. And then by also biopsying guys who stopped using steroids years ago, we're going to be able to try and see, does their muscle still have high numbers of nuclei, and is there a difference? And so that will just add to the body of literature that has come from Christian Gunderson's laboratory. It's come from Anders Ericsson, by just trying to see the nuclei that are incorporated for anabolic steroids, are they retained through time in humans? That will just add to that body of literature. And the unique thing that our project is also going to do that has not been done yet, as far as I know, in the scientific literature, in human actual steroid users using more than physiological amounts. I know it's been done in elderly men using replacement amounts of testosterone and medically supervised amounts of growth hormone, but it's never been done in men who are using super physiological levels of androgens that you would see in the untested bodybuilding, powerlifting, strongman communities, is actually look at the molecular level. What is the difference in both gene expression and epigenetic markers? And some of your listeners might not be familiar with those terms, but in essence, what we're trying to investigate is there a molecular difference inside their muscle in terms of the way proteins are being made? So when a steroid binds to the androgen receptor, it will go into the DNA inside that nucleus, inside the muscle, and it will bind to a very, very specific region, and it will then cause something to be made that's called a transcript, a messenger transcript, and that will then get converted into protein. And that's what everyone's interested in, is the amount of protein. And essentially, we're going to be able to look at that whole pathway. So we're going to be able to see what regions of the DNA are more open and are more closed when someone takes anabolic steroids. Are there differences in the transcripts that are being made? And there are also differences in the proteins that are being made. And the amazing thing that I think will be very unique is if we get enough people in this time course study, where we have people who are just finished a cycle and then they come back 20 weeks later, we're going to be able to see, is there a molecular difference in their muscle in terms of what genes are being expressed and what regions of the DNA are being opened and being closed. Because if the receptor has bound to that DNA and they're taking a large amount of steroids in that time, the DNA will be very opened and will be making certain proteins that the steroid is doing its job for. But the question is, 20 weeks later, is that DNA still open? Are those pathways still being activated even if the steroid has disappeared? Has the muscle exhibited a memory in terms of DNA opening and closing, which is an epigenetic memory of that steroid exposure? And muscle has been shown now last year in 2018 to demonstrate an epigenetic memory of resistance training. So it was a research paper conducted out of Adam Sharple's laboratory. Robert Seaborn was the main author, and they subjected guys to a resistance training program, and they looked at what regions of their DNA were opened and closed for anabolism, for muscle growth. And they actually showed that after a single bout of resistance exercise, certain regions of the DNA get opened for muscle anabolism, for muscle growth. And then they then subjected them to I think it was over a month of consistent resistance training, multiple times a week. And those regions of the DNA got even more opened. Yep. And then they took them away from resistance training, and those regions of the DNA actually closed back up. But they didn't close back up fully. They remained slightly open. And then when they went back to resistance training, those regions of the DNA opened up even further. And so the muscle seemed to exhibit a memory that it was previously exposed to resistance training exercise in terms of what regions of the DNA for anabolism are opened and closed. And we have no idea in humans if the muscle exhibits a memory in the DNA of steroid exposure through time. So there's potentially two memory mechanisms that we could be finding. One is that the myonuclei that are incorporated are retained and mean that protein synthesis will always be higher. And the second one is that the regions of the DNA for anabolism are open and remain open many months after the cessation of usage.
This is fascinating. So this is cutting edge, trying to find out what Mother Nature's intention is. Have you ever considered, last question, Alex, have you ever considered using a super natty, like someone who is just on the genetic outlier, to also study someone, I don't know, that has just attained a level of physique that's not only admirable, but just leaves you wondering? Someone like Michael Hearn, where
yes,
you've had different people. You said sedentary individuals. You want people that are not on. You want people that are on. Have you ever thought about getting a super natty on, just to see how it influences yeah?
That's true. He might be an outlier. I'm not sure. We'll
An
have to
outlier of
see, yeah.
outliers. If we could just study,
Yes.
if we could just study
Yes.
him out, I think we could unlock. It would be the Rosetta Stone of hypertrophy. You know?
Yeah, everyone would just get jacked.
Yeah, that is the dream. No,
The secrets.
I really appreciate you coming back on the channel because I think you did a fantastic job talking last time just about steroid detection. Because it was a topic that, you know, when we exchanged some emails, I didn't know much at all, and then quickly from talking with you, I realized I really didn't know what was going on. But then as it relates to current research and what's actually out there, I think it's very important to try and put out good information, and especially someone that's trying to conduct interesting research, what we know so far, what we hope to know, and what are the practical implications. So I would say, Alex, in closing, is there anything else that you want to say that you feel you haven't covered, anything you want to clarify, anything at all?
No, that's pretty much everything. I'll just say if you are a current anabolic steroid user living in Europe and you're a male aged 20 to 40 years old and you're interested in being monitored for research as you come off anabolic steroids, do get in touch with me. We'll be able to get your travel reimbursed and organize you being able to come to the laboratories. The same goes if you're a past anabolic steroid user who finished taking supraphysiological dosages 2 or more years ago, male aged 20 to 40 years old. As long as you do not have a disorder that would affect your blood to clot, because that would make it unsafe for us to collect your blood or conduct a muscle biopsy, then you can get involved. Don't be concerned about the pain associated with the muscle biopsy. It is considered a minor invasive procedure. I've had a muscle biopsy for this research because obviously it's my PhD. I wanted to see what it was all about, and it really is minimally invasive. There's minimal amount of discomfort associated with it, and the chance of complications are very low. And you will only be taken away from training for 2 to 3 days after the procedure, just to make sure that everything heals up as quickly as possible. So please do get in touch. If anybody out there has also found this conversation incredibly interesting, but they don't fit in the categories of people that we're looking for, you can find a public Facebook post I've made by just searching Alex KT muscle memory. If you shared that, I'd be greatly appreciative. I also have a single Instagram post, Alex_K_T on Instagram. All the details of the study are on there, including the advertisement. I'd greatly appreciate anybody sharing that just to help spread the word, because obviously you can imagine it's difficult to try and conduct this research and recruit people. And that would be a great way for me to be able to spread the word on the research. If you want to get in touch with me, the best way is via my email, akt19@brighton.ac.uk, or my full name @brighton.ac.uk. My email will be in the description box, or contact me via social media, whatever you prefer, and we can then take the conversation forward about getting you involved.
Absolutely. I highly recommend, if anyone's listening, if anyone's watching that can participate, do so, because you stand to help so many individuals. All that information will be linked in the description. Alex, thank you so much for being on the channel. If people enjoyed the video, they could go ahead and like the video, and we'll see all you guys, my rascals, in that next one. Peace.
Eat your vegetables, eat your vegetables, eat your fucking vegetables.